Hello everyone, my name is Leah, and I'll be telling you about the independent project I did at the Dave Schreimer lab at Foothills, where I optimized a protocol to sequence antibodies for the application of CARs and CAR T-cell therapy. CAR T-cell therapy is a cancer immunotherapy where the immune system can be used as a powerful weapon against cancer. The immune system includes specialized cells that can recognize patterns, which are called antigens, like police dogs tracking a scent. And when it finds the pattern, it destroys it and alerts other immune cells to help. CAR T-cell therapy implements this by attaching chimeric antigen receptors, or CARs, on killer T cells that recognize cancer cell patterns, basically training these police dogs to recognize the specific cancer cell and to destroy it. This is done by knowing the sequence of the antibody that recognizes the cancer pattern and inserting it into the cell, for example, by a viral vector, so that the T cell can express it as the CAR. So how do we know the sequence of the CAR? That was where my project came in. It cost $8,000 to sequence an antibody. My project is to be able to do what those antibody sequence companies are doing, but at no cost. I developed my protocol based off of an antibody that targets a cancer pattern that is overexpressed when someone has breast or stomach cancer. This antibody sequence is already known. This interdisciplinary project combines proteomics knowledge using a mass spectrometer with novel breakthroughs in other fields. By finding the right enzymes to digest any given antibody into peptides, which means cutting the antibody in sequence strands, then allowing the mass spectrometer to use its fragmentation methods to give an output spectra showing each peptide's mass-to-charge ratio, we can use softwares that can interpret these spectra to give a peptide sequence. However, this is usually done with a database. But for my project, we don't know the antibody sequence, so we don't have a database. If I was able to have a database, this means the software can compare the spectra to protein sequences we expect to see in our sample and then tell you what proteins are most likely to be present. So without a database, I had to use something called de novo sequencing, where math can be used to figure out the peptide sequence without a database. However, the spectra needs to be very good quality so that there is no ambiguity. I spent a lot of time back and forth in the lab and using the softwares to perfect a bunch of different strategies to create a good quality spectra. These strategies included determining the perfect enzyme combination, digestion times of these enzymes, derivatization, which was a novel idea to add things to the antibodies to make it better to sequence, and changing settings on the different mass spectrometer fragmentation methods. After de novo sequencing, we need another software to align all of the peptides into a full antibody sequence that can be implemented in making CARs. To perfect this aligning, we need a peptide sequence that overlap a lot. And that is why the perfecting of this protocol to give the best peptide pool and good quality spectra is important. I compared my sequence I created with the known sequence found online for the antibody I tested my protocol with, as well as another commonly used cancer antibody. At the end of my 4-month internship, I achieved a 99% sequence accuracy. The 1% of amino acids that were inaccurate were amino acids that have the same mass-to-charge ratio, so it makes it difficult for the softwares to figure out which amino acid it is. Since we know it is one or the other, different sets of CARs can be made and tested for the successful sequence. This is still way cheaper than getting a company to sequence it. Overall, I'm really proud of this project. I'm already being asked back into the lab to sequence antibodies for other labs in the Arnie Charbonneau Cancer Institute. This project is already proving to be an asset for the University of Calgary's cancer research by saving tens and thousands of dollars. Here are my references, and thank you for listening.